based on the latest syllabus. Apart from that, we have personalized counseling sessions, specially curated for you, where we give one-to-one guidance. If you need any extra guidance and support from Auricle, you feel free to book a slot with us anytime. Then we have 3 different subscription plans, that is basic, standard, and mentorship. In mentorship, we have one-to-one personalized guidance and live doubt clarification sessions as well. You can get an additional 20% off if you use my promo code as displayed here on the screen. Briefly telling you about the internal marathon series. This is a rapid revision series. We'll be touching about all the important topics that are frequently asked or could be asked from you. Then we'll talk about the volatile facts and quick revision, and multiple choice questions will also be covered to ensure that you get top-notch grades in your exams. Okay, so in our previous class, we had already got done with glycolysis, gluconeogenesis, glycogenolysis. Okay, so we had covered a major chunk of the carbohydrate cycles. Glycolysis, it was in the well-fed state. Well-fed state after your meals. After meals, when the blood glucose level was increased. Okay, so to normalize, to maintain the blood glucose level, your glycolysis was taking place. Then we had gluconeogenesis. This was in the fasting state to provide blood glucose. Then glycogenesis was glycogen synthesis, and this was also taking place in the well-fed state. What is glycogen? It was the storage form of your glucose. So your excess glucose, which is beyond the means of body to utilize, it was stored for future use in the form of glycogen. What was glycogenolysis? That was the breakdown of glycogen to provide glucose when the blood glucose level would go down. Okay, so today we'll be talking about TCA followed by HMP. Then Rappaport, Cories, and fructose and galactose metabolism. Fructose and galactose metabolism, the metabolism as such is rarely asked, but it has clinical application, so it becomes important. Okay, and as I told you yesterday, that the cycles would be covered under the following heads. That is your cycle of the pathway, organelles, then the state in which it is taking place, energetics, regulation, which is very important, then the clinical, again very important, and in the miscellaneous, any relevant information about that particular topic would be covered. So your TCA is also known as Krebs cycle. Now, I had told you, if you remember in the regular classes, that what is Krebs cycle. In the Krebs cycle, your 2-carbon acetyl-CoA was combining with a 4-carbon oxaloacetate to form a 6-carbon citrate. Okay, so the 2-carbon of acetyl-CoA, it was undergoing oxidative decarboxylation. You can see carbon dioxide release, and this was followed by production of NADH. So it was undergoing oxidative decarboxylation, 2 successive oxidative decarboxylation. So your acetyl-CoA was completely oxidized to form a 4-carbon compound. Now this 4-carbon compound was again via multiple steps changed into oxaloacetate. So your oxaloacetate was regenerated. So I told you, if you divide your Krebs cycle into 2 halves, this could be the first half and this could be the second half of your Krebs cycle. First half and second half. So what was happening in the first half? In the first half, it was complete oxidation of acetyl-CoA. Okay, and it was completely oxidized by being converted into carbon dioxide molecules. So 2 carbon dioxide molecules were liberated. And in the second half, what was happening? The second half was basically regeneration of oxaloacetate. So therefore it is sometimes also said that oxaloacetate works as a catalyst in Krebs cycle because it enters Krebs cycle and it is regenerated again and comes out the same without being unchanged, without being changed. Okay. Now moving on to the steps of TCA. So you can see here this oxaloacetate, although the structures are shown in the figure, but you need not know the structures. Writing the words is more than sufficient, okay? So oxaloacetate, it combines with acetyl-CoA and the enzyme is citrate synthase. And what is what? Citrate is formed, okay? Now this citrate is acted upon by ecotinase enzyme to form what? This is a two-step reaction. First, an intermediate cis ecotinate is formed, which is later on converted to isocitrate. So directly speaking, your citrate, it forms isocitrate by the action of the enzyme ecotinase. And I told you this was a moonlighting enzyme. What are moonlighting enzymes? They are those enzymes that apart from the enzymatic activity function, they have some other function as well. So this ecotinase, it was involved in iron homeostasis, okay? Now this isocitrate is acted upon by the enzyme isocitrate dehydrogenase to form what? To form oxalosuccinate. Okay. Now this succinate, it is acted upon by isocitrate dehydrogenase. This is again a multi-step reaction and to form alpha ketoglutarate. So you can skip this step as well and directly write that your isocitrate is acted upon by isocitrate dehydrogenase to form what? Alpha ketoglutarate. Now this alpha ketoglutarate, it forms succinyl-CoA and the enzyme is alpha ketoglutarate dehydrogenase. Now this succinyl-CoA is acted upon by succinate thiokinase to form succinate. This succinate is then changed into fumarate by the action of enzyme succinate dehydrogenase. Now this fumarate changes into malate by the enzyme fumarase and then malate again forms what? Oxaloacetate by the enzyme malate dehydrogenase. So this is the Krebs cycle and you need to remember the substrates, the products forms and the enzymes involved for each of the steps. Apart from that, what is important? You need to remember in which of the steps decarboxylation is taking place, okay? That is your carbon dioxide is being removed. So here one carbon dioxide is being removed and here one carbon dioxide is being removed. So these are the two steps where your carbon dioxide is being removed and then you also need to know in which of the steps NADH is formed. So here one NADH is formed, here one NADH is formed. Then also one GTP or ATP is also formed, okay? Here you can see. Apart from that, one FADH2 is formed in this step and one NADH again is formed here. So you need to know in which steps the reducing equivalents are being formed, the carbon dioxide liberated in which steps, and apart from that, this substrate level phosphorylation that is taking place, one ATP being formed. Now, what is the site? All organs with mitochondria perform your TCA. Organelle is mitochondrial matrix, except for succinate dehydrogenase, which is a part of complex 2 of electron transport chain. So this is present in inner mitochondrial membrane. So apart from your succinate dehydrogenase, rest of the TCA enzymes, they are present in the mitochondrial matrix and it takes place both in the fed and the fasting state. Therefore, there is no hormonal regulation for your TCA. Because it is taking place both in the fed and the fasting state. Now moving on to the energetics, you have 3-NADPH being formed in TCA. What are the formed here? The other NADH, second NADH formed here and one NADH formed here. So 3-NADH being formed in your TCA. Now this 3 NADH, it will enter into electron transport chain. 1 NADH gives 2.5 ATP. So 3 NADH will give 7.5 ATPs. 1 FADH2 is also being formed, and 1 FADH2, when it enters into electron transport chain, it gives 1.5 ATP. Apart from that, there was one substrate level phosphorylation. So 1 ATP formed here. So a total of 10 ATPs are being formed from the run of one TCA cycle. So one TCA cycle, it produces 10 ATPs. Okay, so you need to know the energetics. Now moving on to the regulation of TCA. So what are the regulatory enzymes? They are citrate synthase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, and pyruvate dehydrogenase. Although this pyruvate dehydrogenase is not a part of TCA, this is the link reaction. What is the link reaction? From glycolysis, glucose, when it undergoes glycolysis, it forms what? It forms pyruvate. And then that pyruvate, it forms acetyl CoA, and this is by pyruvate dehydrogenase, and this is the link reaction. So this link reaction is not considered as a part of TCA, but this is responsible in regulating TCA. Okay, not a part of TCA, but it regulates TCA. Now, high energy states inhibits TCA. So when there is already high ATP in the body, that is high ATP ADP ratio, then your TCA will be inhibited, because what is ultimately TCA forming? It is forming ATP. Okay, when the body already has enough ATP, there is high ATP in the body, it will inhibit TCA. High NADH NAD ratio will also inhibit TCA, because NADH is ultimately going into the electron transport chain and giving you further ATPs. Then isocitrate dehydrogenase is inhibited by NADH and ATP. All dehydrogenases in the TCA are favored by increased level of calcium in muscles. So when your calcium levels are increased, the dehydrogenases of the TCA, they are favored. That is, their activity is enhanced. In the brain, the TCA is regulated at the pyruvate dehydrogenase level. That is, this is your link reaction. So in the brain, your link reaction is controlling the TCA. There is no hormonal regulation of TCA, as TCA should happen whether the body is in fed or the fasting state. So there is no role of insulin and glucagon in the regulation of TCA. Now moving on to the inhibitors of TCA, fluoroacetate, which non-competitively inhibits equinase. Okay, then arsenite, this inhibits alpha ketoglutarate dehydrogenase, and your malonate, that inhibits succinate dehydrogenase. So these are the 3 inhibitors of TCA. That is your fluoroacetate, your arsenate, and your malonate. Now, one of the important roles of TCA is its anaplerotic role, that is filling up reactions. These are filling up reactions. Okay. So if you see, if your oxaloacetate is depleted, it will be filled up by aspartate. Now in the cycle, if your alpha ketoglutarate is being depleted, then your glutamate, histidine, arginine, proline, and ornithine fatty acids, they will replenish the alpha ketoglutarate. If your succinyl coenzyme A is being depleted, it is being replenished by valine, isoleucine, and methionine. And your fumarate, it is being replenished by phenylalanine and tyrosine. If you remember the metabolism of phenylalanine and tyrosine or the other amino acids, then you will know the ketogenic and the glucogenic amino acid that we studied, how fumarate was being formed in phenylalanine and tyrosine metabolism in the air. So it was replenishing fumarate here in the TCA cycle. So these filling up reactions, that is the replenishment of the substrates of the TCA, that is known as anaplerotic reactions. So that was briefly about TCA. So for all the cycles, you need to know the cycles as such. That is the product substrate in the enzyme. In which step your NADH is being formed or ATP is being formed or utilized or a carbon dioxide being liberated, you need to know that as well, followed by energetics and regulation. Now moving on to the HMP. So we've covered glycolysis, gluconeogenesis, glycogenolysis, and TCA. Now we'll be talking about HMP. If you're finding it hard to follow at this pace, then I would recommend that you go back and listen to the regular lectures where everything has been explained in detail. The purpose of this series is only to touch upon the periphery and brush up what we've already studied. Okay, so this is simply a revision class and we're trying to cover major chunk of the syllabus. Okay, so we'll prefer not going into the detail. But if you want to hear the lecture in detail, then you are free to anytime listen to our already recorded lectures that are available. Now moving on to HMP shunt. So HMP shunt, it has two phases. That is your oxidative phase and non-oxidative phase. Your oxidative phase is irreversible and your non-oxidative phase was reversible. So your HMP shunt, that is hexose monophosphate shunt. So the glucose, instead of entering glycolysis, it is shunted into this pathway. And this pathway is composed of two phases, that is oxidative phase, which is irreversible, and non-oxidative phase, which is reversible. Now, what is happening in the oxidative phase? Your glucose 6-phosphate. That is your glucose forms glucose 6-phosphate. Now, this glucose 6-phosphate, it is acted upon by glucose 6-phosphate dehydrogenase, which is the rate-limiting enzyme, and it forms ultimately what? Ribulose 5-phosphate is formed with one carbon dioxide being liberated and two NADPH molecules being formed. So, basically, in the oxidative phase, you need not know the structures. The minimum that you should know is that your glucose 6-phosphate is acted upon by the enzyme, that is glucose 6-phosphate dehydrogenase, to form what? Ribulose 5-phosphate. And in this process, one carbon dioxide molecule is liberated and two NADPH is being formed. So if you write this also, this equation that I've written here, that is also more than enough. So that was about the oxidative phase. Now moving on to the non-oxidative phase of HMP. In the non-oxidative phase, what happens? Ribulose 5-phosphate that was formed in the previous phase, three of them, you can see, three molecules of ribulose 5-phosphate, okay? So one, by the action of epimerase, it forms what? It forms xylulose 5-phosphate. The other one, by the action of ketoisomerase, it forms ribose 5-phosphate. And the third one, it also forms xylulose 5-phosphate. Now this ribose 5-phosphate, this is going to go for synthesis of nucleotides, that is RNA and DNA. So what is the significance of your HMP? It is not used for the production of ATP. So energy generation or ATP production is not the aim of HMP pathway. Rather, to provide NADPase is the aim of your HMP pathway, and also to provide ribose 5-phosphate pentoses for the synthesis of RNA and DNA. So that is the significance of HMP. Now your xylulose 5-phosphate and your ribose 5-phosphate here, they are acted upon by trans-ketolase enzyme to form what? Glyceraldehyde 3-phosphate and pseudoheptulose 5-phosphate. So from xylulose, which is 5 carbon, 2 carbon is transferred to ribose 5-phosphate to form pseudoheptulase. This is 7 carbon, okay? And what is left here is 3 carbon glyceraldehyde 3-phosphate. Now this glyceraldehyde 3-phosphate and pseudoheptulose, they are acted upon by trans-aldolase to form what? Fructose 6-phosphate and erythrose 4-phosphate. This erythrose 4-phosphate is 4 carbon. So from this pseudoheptulase, which was 7 carbon, 3 carbon are transferred to glyceraldehyde to form a 6 carbon fructose. And what is left behind is erythrose 4-phosphate. Now this erythrose 4-phosphate, which is left behind, this is acted upon by trans ketolase. And what is the other substrate? The third xylulose 5-phosphate. Okay, that was there. That is the next substrate to xylulose 5-phosphate and erythrose 4-phosphate. They are acted upon by trans ketolase to form what? To form fructose 6-phosphate and glyceraldehyde 3-phosphate. So we see we have one fructose 6-phosphate and other second fructose 6-phosphate. Now this glyceraldehyde 3-phosphate changes into fructose 1,6-bisphosphate, but half is written because fructose is 6 carbon and glyceraldehyde is 3 carbon, so that is half a mole. Okay, and then these are reversible reactions. Okay, this oxidative, non-oxidative phase, this is reversible. So depending upon the need, it can either form the intermediates of glycolysis or it can form pentoses. So depending upon the requirement of the body, these reactions will take place. Either they will move in the direction of the formation of glycolytic intermediates or in the formation of pentoses, because all of these reactions are reversible. Now the site, that is the organs where your HMP is operating. So your irreversible phase, the oxidative phase, it is particularly important in those organs where there is active NADPH dependent biosynthesis of fatty acid. So in fatty acid synthesis, you have studied what is needed. NADPH is needed. Okay, so those organs where there is active fatty acid synthesis taking place, so NADPH will be required. So particularly in those organs, what becomes important? Your irreversible oxidative phase, it becomes important. So which are those organs? That is your liver, lactating mammary gland, and your adipose tissue. Now, they are also important in those organs where there is NADPH dependent biosynthesis of steroid hormones taking place. Those are your testes, ovaries, placenta, and adrenal cortex, where your steroid hormones are being synthesized. And in the RBCs, which need NADPH to keep glutathione in reduced state. Okay, so your glutathione, which is important in free radical scavenging, and for it to scavenge free radical, it needs to be in the reduced state. And what is responsible for keeping it in its reduced state? You need NADPH. Okay, so it is also important in RBCs. Now your non-oxidative reversible phase occurs in almost all tissues to produce pentoses, as DNA and RNA is synthesized in all the organs. So your reversible phase is taking place in almost all the tissues, because every tissue will need to synthesize DNA and RNA. And this is a cytoplasmic pathway. Now moving on to the regulation, the glucose 6-phosphate dehydrogenase is the rate-limiting enzyme involved in the first step of oxidative phase. It is inhibited by NADPH. Okay, so if you have excess NADPH, it will inhibit glucose 6-phosphate dehydrogenase. High levels of NADPH inhibits glucose 6-phosphate dehydrogenase, and low levels enhances its activity. So one of the important regulators of glucose 6-phosphate dehydrogenase is NADPH. Remember that. Then insulin, it upgrades the expression of the gene for glucose 6-phosphate dehydrogenase. Okay, so that is insulin induces glucose 6-phosphate dehydrogenase and thereby increases the overall pathway. So your insulin is activating what? Glucose 6-phosphate dehydrogenase. So in the presence of insulin, your HMP will be enhanced. Then non-oxidative phase is controlled by requirement of pentoses. So in those organs like in the intestine and the skin, where there is a lot of wear and tear, so there needs to be more repair. So for that you need more DNA synthesis. So in those organs where there is more requirement of pentoses, your non-oxidative phase will be working more. Now clinical, this is very important, glucose 6-phosphate dehydrogenase. This is important and you can get asked about it. So it is an X-linked inheritance disease. It is the most common disease-producing enzyme abnormality in humans. What is defective here? There is decreased glucose 6-phosphate dehydrogenase. Now, decreased glucose 6-phosphate dehydrogenase means there is decreased NADPH, because when your HMP is not working, you will not have NADPH. And When your NADPH is not there, so all the functions that are related to NADPH, they will be hampered. Increase free radicals. As I told you that NADPH is involved in free radical scavenging by keeping your glutathione in reduced state. So if there are no NADPH because of reduced HMP, what will happen? There will be increased free radicals, and we all know how damaging the free radicals are. Then drug-induced hemolytic anemia and favism. That is in fava beans. If you consume them, you get favism. Then Wernicke-Korsakoff syndrome, that is due to thymine deficiency. How is thymine deficiency related here? Let's go back. So you see this trans-ketolase, this needs thymine. Okay, this is a thymine-dependent enzyme, this trans-ketolase. So in case of thymine deficiency, you get Wernicke syndrome. Now, in miscellaneous, you have the uses of NADPH. Okay, it is important that this uses of NADPH, basically, this is also the significance of HMP. So either you can be asked that what is the significance of HMP or the uses of NADPH. So we know it is involved in reductive biosynthesis of steroid hormones, cholesterol, and fatty acid. Then it is involved in free radical scavenging. Free radical scavenging is very important in the body because we all know about the harmful effects of free radicals, how damaging they can be for the bodily proteins. So it is very important for the body to detoxify them and scavenge them. Then lens transparency, if free radicals accumulate, then your lens will become opaque. Then RBC membrane integrity is also maintained by NADPH. Then it prevents the formation of methemoglobin. This is also very important. If there is more methemoglobin, then the transport of oxygen will be hampered. Then macrophage bactericidal activity. It is also dependent on NADPH. So that was briefly about your HMP pathway. Now moving on to Cory's cycle. So what is happening in Cory's cycle? That in the muscle, especially in strenuous muscles or actively exercising, what is happening? So glucose, it changes into pyruvate. And due to the absence of oxygen under anaerobic conditions, that is when the muscle is rapidly exercising or in the RBCs, where your anaerobic glycolysis is taking place, what is formed? Lactate is formed. Now, to prevent this lactate accumulation, what happens? This lactate goes to the liver via the blood. Now, what is happening in the liver? This lactate is changed into pyruvate, and then this pyruvate forms glucose. So, this is happening via gluconeogenesis. And this is what is known as Cory's cycle. And then this glucose via blood is again transported back to the muscle. So the transport of lactate from the muscle, that is the lactate formed in the muscle, its transport via the blood into the liver, and then that lactate serving as a substrate for gluconeogenesis to again form glucose, and then glucose is again transported back to the muscle. This cycle is known as Cory's cycle. So now you know what is Cory's cycle. Cory's cycle is in the muscles, that is from the RBCs or actively exercising muscles, the lactate that is formed, it is transported via the blood into the liver, and in the liver then it undergoes gluconeogenesis, serves as a substrate for gluconeogenesis and forms glucose again, and then that glucose is again transported back to the liver, and this entire thing is known as Cory's cycle. Now what is the significance of Cory's cycle? Its significance is to prevent lactate accumulation in the body. Okay, and why do you want to prevent lactate accumulation? Because this will lead to muscle fatigue. So in order to prevent muscle fatigue, we need to prevent lactate accumulation. So for this reason, the body utilizes Cory's cycle. So this was about Cory's cycle briefly. Actually, this is all you need to know, and this is all what the Cory's cycle is. And you might get asked about your short notes. Then you have Rappaport Lubering cycle, that is your BPG shunt. Now, if you remember in glycolysis, the sixth step of glycolysis, what was happening there? Your 1,6-bisphosphoglycerate that was formed, it was changing into 3-phosphoglycerate. And the enzyme involved was bisphosphoglycerate kinase. This is the only reversible kinase. If you remember, I told you this. Only reversible kinase. So what was happening in this step? The 1,3-bisphosphoglycerate was being acted upon by bisphosphoglycerate kinase, and it was changing into 3-phosphoglycerate, and this step was releasing one ATP. So there was substrate level phosphorylation taking place. Okay, so you were obtaining one ATP. Now what happens in BPG shunt, that this conversion of 1,3-BPG into 3-phosphoglycerate, this is taking place, but the mechanism is different. The enzyme is different. How? This 1,3-bisphosphoglycerate is not acted upon by this bisphosphoglycerate kinase. Rather, it is acted upon by mutase to form what? 2,3-bisphosphoglycerate. So what is this mutase doing? That within the enzyme, it is causing transfer of phosphate. In 1,3-bisphosphoglycerate, there was phosphate as first carbon and there was phosphate as third carbon. You can see here. Okay, now what is mutase doing? It is transferring this phosphate from first carbon to the second carbon. So this is 2,3-bisphosphoglycerate. So there is phosphate at second carbon and there is phosphate as third carbon. Now what is happening? It is acted upon by phosphatase, and we all know what do phosphatases do? They remove phosphate. So phosphatase, it removes the phosphate from position 2 and you get 3-phosphoglycerate. So in this BPG shunt, you are again getting 3-phosphoglycerate, but the enzymes used is different. This step of substrate level phosphorylation, this is bypassed. So in BPG shunt, no ATP produced. But in the sixth step of glycolysis, you can see ATP was produced. There was substrate level phosphorylation taking place. So this bypass of the kinase enzyme, this is what is known as your BPG shunt or Rappaport Lubering cycle. Now this takes place in mature RBCs. That is, your BPG shunt is taking place in the mature RBCs. Now, what is the significance of this? The significance is that your 2,3-BPG that is formed, the intermediate that is formed, it reduces the affinity of hemoglobin for oxygen. Therefore, it helps in unloading of oxygen more easily in the tissues. So basically, your 2,3-BPG that is formed, it is helping in unloading of oxygen. Now, under hypoxic conditions, the 2,3-BPG concentration increases in the RBCs, thus favoring the release of oxygen even in low, when the partial pressure of oxygen is low. So, whenever there is condition of hypoxia, the partial pressure of oxygen falls down. So, the unloading of oxygen becomes difficult. But what is happening when there is increased 2,3-BPG? Then in that case, what is the 2,3-BPG doing? It is decreasing the affinity of hemoglobin for oxygen, thereby enabling unloading of oxygen. Thus, it becomes important in hypoxic conditions. That is, your 2,3-BPG, it reduces the affinity of hemoglobin for oxygen and therefore it helps in unloading of oxygen. Now, your 2,3-BPG is also increased in high altitudes. Again, in high altitudes, the partial pressure of oxygen is low. Therefore, there this 2,3-BPG becomes important, and in fetal circulation. So, this is the significance of your BPG shunt, and this is important. Now, moving on to fructose and galactose metabolism. So, we've covered almost the entire cycles of carbohydrate metabolism. And also with this, we have covered a very important and major chunk of carbohydrate metabolism. Glycolysis done, gluconeogenesis done, glycogenolysis done, TCA done, HMP done, Rappaport done, Cori done. So we've completed all these cycles. Now moving on to fructose and galactose metabolism. So fructose metabolism, what is happening to fructose? Whatever you get from diet, this fructose is acted upon by the enzyme fructokinase to form what? Fructose 1-phosphate. Now, this fructose 1-phosphate is then acted upon by the enzyme aldolase to form glyceraldehyde and dihydroxyacetone phosphate. Now, it is acted upon by another enzyme, that is triose kinase, which phosphorylates glyceraldehyde to form glyceraldehyde 3-phosphate. And now, this can enter glycolysis. So what is happening in fructose metabolism? The initial two bottleneck steps of glycolysis, that is the hexokinase and phosphofructokinase, that is being bypassed. Okay, so this fructose metabolism increases the flux for glycolysis. Therefore, we see that fructose is readily metabolized. Now, this is very important. The clinical related to fructose metabolism. It is very rare that you might get asked about fructose metabolism as such, but this is something which you get asked. Hereditary fructose intolerance. Now, what is it? It is an autosomal recessive inborn error of metabolism, and the defect is in aldolase B. Here, this enzyme aldolase B, if this is absent in hereditary fructose intolerance. So what will accumulate? Your fructose 1-phosphate will accumulate. Seen when sucrose is introduced in the diet of infants, usually around the 6 months of age. So when weaning starts and you introduce the baby to food, what is your sucrose? Your sucrose is the disaccharide which contains glucose and fructose. So that is when the baby is getting fructose for the first time. So in that case, what happens? This is manifested. Then accumulation of fructose 1-phosphate inhibits glycogen phosphorylase. Now, what is glycogen phosphorylase? If you remember, this was the rate limiting enzyme of glycogenolysis. So this leads to the accumulation of glycogen in the liver. So if glycogen is accumulating, what will happen? Hepatomegaly will happen. See here one of the manifestations. And also if your glycogen is not being broken down, it is accumulating, it will lead to hypoglycemia. Okay. Now, other clinical manifestations include vomiting, loss of appetite, hepatomegaly as discussed, jaundice, there will be liver damage and failure to thrive. Then withdrawal of fructose from diet will immediately relieve the symptoms. So the treatment is that you withdraw fructose from the diet. Now, what is fructosuria? It is due to deficiency of fructokinase. What was fructokinase? It was this enzyme which was converting fructose into fructose 1-phosphate. So if this is absent, it will lead to fructosuria. There is no abnormality other than excretion of fructose in urine. Now moving on to galactose metabolism. You can see this galactose that you have either through diet or it is also formed in the body from UDP glucose. So this galactose, it is acted upon by the enzyme galactokinase to form what? Galactose 1-phosphate. Now this galactose 1-phosphate, it combines with UDP glucose in the presence of the enzyme, that is galactose 1-phosphate uridyl transferase. This is the rate limiting enzyme, and what will happen? Your UDP glucose, it will form UDP galactose, and your galactose 1-phosphate, it will form glucose 1-phosphate. Okay. Now, what is happening? This UDP galactose that is formed, it is acted upon by the enzyme epimerase to form UDP glucose, and this UDP glucose then it can contribute to glycogen synthesis. Okay. Now what is the fate of this UDP galactose? This UDP galactose is also formed from galactose phosphate via the enzyme galactose 1-phosphate pyrophosphorylase. So that is about your galactose metabolism. So basically your galactose, it is acted upon by galactokinase to form galactose 1-phosphate. Now this galactose 1-phosphate, it combines with UDP glucose to form UDP galactose. And this UDP galactose, it is also involved in lactose synthesis. And also this UDP galactose, the other fate is that it can be acted upon by the enzyme epimerase to form UDP glucose. And we all know what is the function of UDP glucose. It provides glucosal residue in glycogen synthesis. Now, galactose is metabolized almost exclusively by the liver, and therefore galactose tolerance test is done to assess the functional capacity of the liver. So this is one of the asked MCQs. Now, clinical associated with galactose metabolism, again, this is important, galactosemia, deficiency of enzyme galactose 1-phosphate uridyl transferase. So the enzyme deficient is this one here. This is defective. You can see I am first galactosemia. It is again an inborn error of metabolism. Galactose 1-phosphate will accumulate in the liver, thus inhibiting galactokinase, and it also inhibits glycogen phosphorylase. Again, the glycogen phosphorylase is inhibited. What will happen? Hypoglycemia will occur because glycogen phosphorylase is involved in glycogenolysis, that is breakdown of glycogen. So that will not take place. So glycogen will start accumulating, leading to hypoglycemia. There is liver enlargement, that is hepatomegaly due to accumulation of glycogen. Then there is jaundice and severe mental retardation. Now, another characteristic feature of galactosemia is your congenital cataract. Why? Because your galactose, it can also be reduced to dulcitol, and this dulcitol, when it accumulates in the lenses, it causes cataract. Now, galactose 1-phosphate, it may also get deposited in renal tubules, so it can also cause, apart from liver damage, it can also cause kidney damage and brain damage as well. Now, producing tubular damage leading to generalized amino acid uria. A treatment includes lactose-free diet. Okay, so that was briefly about your galactose and fructose metabolism. So today we come to an end of all the cycles in carbohydrate metabolism. Fructose metabolism also done and galactose metabolism also finished. So with this we come to the end of the cycles. Almost the major and important portion of carbohydrates have been covered. Now we left with chemistry and gas and a few other topics, which we'll take in the subsequent classes. If you have any queries, you can ask. And if you want to hear to the lectures in detail, then you can anytime go back to our routine classes where everything, all these topics, they have been taken in detail, and our lecture on all these cycles. Okay, this class is only meant for the purpose of brushing up what we've already done. Any queries you can ask? Okay, so with this we come to the end of the class. Thanks for watching.